So it's 4:24. I'll wait another minute. You can all get ready, including me. So this is Enzyme Lab Part 2. Part 2. Continuing from last week. My markers, get those out. All right. All righty. So this is Enzyme Action Part 2 lab. And you'll find a lot of similarities this week with last week's lab. So the concept with breaking down the starch, using Lugol's iodine, the 96 well plate. A lot of things are going to stay the same this week, but we're also going to do something different. And by that, we're going to be testing different environmental factors. So for my synchronous section that's watching right now, because I send this to the asynchronous section, we'll be lecturing on enzymes very soon. And we'll be looking at the different environmental factors that can influence enzyme activity. And that's what part 2 is devoted to. It's observing different environmental effects on enzyme function, how enzymes can be really altered by different environmental conditions such as the pH, temperature, as we'll see in this lab, even things like salt, NaCl, so the salinity, we call it, of the environment. And what can happen or not happen is, as we've talked about, this theme of structure and function being intimately related and very much connected. If you mess with the structure, you mess with the function. And we're going to see that certain environmental characteristics or certain environmental conditions, such as excessive heat or acidity with pH, will affect enzyme structure, the structure of the protein, and therefore the function, which can be pretty bad. If these enzymes, which all of them are very useful in our cells, so we don't want that. But it's important to understand what's going on so that we can, monitor it and learn about it. So in this lab, we use the optimal saliva dilution that we discovered last week. And I believe from Stacey's videos and her data collected, it's 1:16 was determined to be the optimal dilution. Okay. So the 1:16 dilution factor was our optimal saliva dilution from last week. I'll add optimal saliva dilution. So for this lab, we're going to want to go ahead and immediately prepare a test tube of optimal saliva diluted to this optimal concentration. So we're going to collect saliva again, fun. And to make this, we're going to get a fresh test tube, right? And we're gonna make this optimal dilution just right away. Remember last time we created a serial dilution, we had a bunch. We're just gonna create our optimal one right off the bat. And so since it's 1 to 16, we're gonna have 15 milliliters of water and 1 milliliter saliva. And that gives us a 1 to 16 ratio saliva to the overall volume, right? And so now, and you know, you mix it, do a little light tapping and mix it up. So we've got our optimal saliva dilution in a test tube there. Cool, got that. Okay, so in this lab, like I said, we're testing different environmental factors. Namely, I believe 3 total. 3 total. And I sent you the packet info, so you should have this in front of you, right? 3, pH, NaCl, which is sodium chloride or salt, and temperature. So what I'm going to do for this lab, since we're not in person, usually I'd assign lab groups a different factor to test. I'd assign a couple groups pH, I'd assign a couple groups temperature to do and test throughout the lab. I'm going to let you guys choose. Just let you choose. I'm not going to assign it. And just choose one. Choose one you want to, you know, learn about and look at. And Stacy has pictures of data and things for each one in the lab. So what I'm going to do in this lab mini lecture is I'm going to go through each of the 3, how to run the experiment for each of the 3 and test each of the 3. And then you can choose one based on which one you're curious about. And so if you chose NaCl as your environmental factor, you would complete this page, but you wouldn't complete the page for temperature or pH. However, you will, it does say everyone must respond to the following. I do want you to learn, right, about the results and effects of all the environmental factors. So you will have to answer this page on all of them, just by looking at the data. But as far as graphing and, you know, analyzing the data collected to, oh, I get what she's doing. I didn't assign you a hypothesis last week. So you can come up with one. Based on what environmental factor you choose, let's say you chose NaCl, or you choose NaCl, your hypothesis could be, for example, increasing the concentration of NaCl will result in a decreased level of enzyme activity. And then for that hypothesis, you look at the data that Stacy collected for NaCl and just write for that question, oh, the data support my hypothesis. It seems like it's true, it's valid. And then you would graph. You only have to do that for one factor. But again, looking at all the data for all the 3 factors, I do want you to answer the questions right before the post-lab questions on all 3 so that you can understand how these factors will affect enzyme activity. And then, of course, the post-lab questions I want you to answer as well. Okay. But I want to go through how we're testing these things and how Stacy has it set up, why the data look like they do with the 96 well plates. So I'm going to start with NaCl. Get my paper here. Okay. All right, so we've got our 1 to 16 optimal saliva dilution. We have a test tube of that. So I'm just gonna erase this. First part here is gonna be for NaCl, if you choose that. And just keeping in mind, we have a test tube put aside with our 1 to 16 dilution here. And this is how you're gonna set it up, okay? You're gonna have, where is it? Okay. What you're gonna have is a positive control and a negative control. The reason for this is you want to make sure your chemicals, your reagents are working properly, and they also give you a point of comparison, and I'll explain that. But for your negative control, let's start with that. Let's do negative control. Do here, and then you get a positive control. So the negative control is going to have water, one milliliter to be exact, 1 milliliter of water. Come on. Okay, cool. 1 milliliter of water, 2 milliliters of starch, and 2 milliliters of the pH 7 buffer from last week. That'll be your negative control. So water, starch, and buffer aren't gonna react, they're not gonna do anything, so you expect a negative result, right? The positive control is going to have okay, sweet. A milliliter of your 1 to 16 saliva dilution, and then 2 milliliters of starch, and 2 milliliters of pH 7 buffer. Okay. Just double checking that they're all the same. Okay, perfect. So that's how you do a negative and positive control. And the negative control, again, nothing's happening, but the positive control, you have your enzyme amylase in your saliva, right? And that's gonna react with your starch. And you expect that. You found that out last week. So that's why it's a positive control. And you do this because if your positive control, let's say, doesn't come out positive, you don't see any enzyme breakdown. of the amylase, then maybe it's you forgot to add something and your technique is off or something. So controls are good for that. They're ensuring that maybe the buffer is working properly, things like that. And again, it's a point of comparison too. And so when you have your negative control and you add it to your well plate, no starch is going to get broken down. So you're going to expect to see all purple, right? Or dark blue. You're not going to see any breakdown because you didn't add any enzyme. And so you can kind of use it as a point of comparison for when you do see breakdown with your enzyme. Okay. So for the actual experiment, and also your positive and negative controls for every environmental factor, it's going to be the same. Same sort of setup here. So I probably won't do it every time. Now, for setting up the NaCl experiment, so I'm going to write, that's annoying. You guys hear that? All right, so the experiment, you're going to test, right, the enzyme activity in different conditions with NaCl. So what that would mean is differing concentrations of salt, and I believe, let's see how much in this one. How many, so Stacy has a 1% NaCl, a 5%, 10%, 20%, and 30% that she used. But I'll just show you one example. We'll try the 1% NaCl. And then you would replace it for each differing condition of NaCl concentration. But let's just do an example for 1%. And this is how you would set it up. So you get a new test tube. And what you're going to do, key for this lab, is you want to subject the enzyme to the condition before you add the starch and allow it to react. You want the enzyme to be exposed to either the temperature that you're testing, the pH condition you're testing, the NaCl concentration, and then see if the enzyme is as efficient or if it's working as well, or you want to test that. So what you do is you subject your enzyme that's present in your saliva. So you can take a milliliter of your optimal saliva from your tube, so 1 ml saliva, put it in this test tube, and then you're going to subject your amylase enzyme in here to, what did I say, 1%? 1% NaCl, and we'll do 2 milliliters. Then you're going to let that chill. You're going to let the enzyme be exposed to that salt concentration for maybe 7 minutes. Okay? And if you remember last week, where we had the starch in the tube first, right? And then we were adding the saliva to it. And then we would wait the 30 seconds. And then after the 30 seconds, you would pipette into the wells. And then another 30 seconds pipette, another 30 seconds pipette. This time, we're not adding the saliva in. We're letting the saliva be exposed to the environmental condition we're testing. And no reactions taking place, right? The starch is our substrate for the enzyme. So after about 7 being exposed to the 1% NaCl, then we add the starch. So after the 7 minutes, we're going to add, so then add starch. And we'll add 2 milliliters. So 2 milliliters of starch. And then, as we learned last week, as soon as the starch and enzyme are in contact, that reaction is going to start happening. So right after you do it, you got to wait 30 seconds, right? And then do your pipetting, same as last week, same sort of thing. So you wait 30 seconds, and then you would pipette 100 microliters. And remember, we would have our 96 well plate already set up. This is just a rough representation. It would be already set up with 100 microliters of that Lugol's inside each of the wells. Oh, should have had my orange marker ready. It's like a pale yellow. And I should also mention, you'll see this when you look at Stacy's data. The first row, she does either the positive or negative control. What does she do? Oh, she does it on the first page. So never mind. Okay. But as far as the negative control, no enzyme present, right? It was just water. So that's a good negative control. There's no enzyme in the water. And it would all turn black. No starch being broken down. That's a good negative control. And then in the second row, the positive control is just what we did last week. It's the saliva. You're just at room temperature like last week, and you expect this gradual breakdown of the starch. So the color change until the 3 wells didn't change colors, right? But then the rows further down, you're going to try this. After exposing enzyme to the condition, adding starch, you're going to do the 30 seconds after that. And the key here is that you'd have a row for your 1% NaCl, and then after you did a row with that, and this 30-second intervals, you'd replace it with, what is she at, 2%? 5%. So you try the same thing next on the next row. So if this was your first one, your first experimental condition, you would try the next row with another test tube, but this time with 5% NaCl, exposed to the enzyme in saliva for 7 minutes, and then you'd add your starch. Wait 30 seconds, pipette, 30 seconds, pipette. In the row below that, and so on and so forth for the higher percentages available. And you'll learn, as the enzyme's exposed to higher levels of NaCl, how that's affecting the reaction. Is more starch being broken down or less in these higher salt conditions? That's the key. And so now that you kind of understand the process, I'm just going to quickly walk through the other ways to do this with the other conditions. That's how you would do it with NaCl. You'd have the same controls for the next other two. So let's do, we did NaCl, let's do pH. Let's do pH. So varying the pH instead of the NaCl. So I'm not going to do the controls again, but you'd have the same positive and negative controls with water and then just your optimal saliva, you expect to see all blue-black for negative control. And then between, I believe, what was it, 4 to 10, you'd see the 3 consecutive no color change in the wells for your positive control, where you have starch in your enzyme. Then below that, you would try screwing around with the pH, see how that affects enzyme activity. Enzyme activity, of course, being the starch breakdown. And whether or not the amylase is going to catalyze that breakdown of starch or hydrolyze that starch. Okay, so for pH, let's do, she has pH 3, pH 5, 6, 9, and 11. So let's just do pH 5 for our example. But again, we have multiple test tubes for each one, each condition. So let's try pH 5. And it's similar to before with the NaCl. You're going to have your 1 to 16 saliva, right? And you would have your 2 milliliters of pH 5 solution, and then and this is 1 milliliter, right? And then same sort of deal, letting it incubate. About 5 minutes for this one. Let the enzyme be exposed to that pH. And then the starch, 2 mils starch, you would add. And the reaction would occur immediately. You would wait the 30 seconds and then pipette into the row. And then for the other columns, rows, you would replace pH 5 with the other pH solutions that we have in the lab and do 2 milliliters of pH 7, pH, sorry, I'm just gonna turn that off. pH whatever. Got pH 6, 9, 11. She has the data for it. So that's how you do it with pH. Same negative, positive controls. Last but not least is temperature and subjecting your tube to different temperatures. All over the place today. Ugh, jeez. Okay. All right. So let's do an example. We'll do the 0 degrees freezing temperature, 0 degrees Celsius. So for the temperature experiment, what you're gonna do is have your enzyme, your 1 milliliter of your saliva, optimal saliva. And interestingly enough, you're also gonna add, just to keep things, the only variable, right, we've learned about this with scientific method, you want the only variable that you're testing for to be temperature, right? That's what you're looking at. So you don't want pH to influence at all. So you're actually just going to add neutral pH buffer. So this is, I think it's pH 7, right? Yeah. So you'd add 2 milliliters of neutral pH solution to your experimental test tubes for temperature. Okay. Cool. Yeah, and you didn't have to do that for NaCl, I don't believe so. But you'd add the buffer for the temperature one. Then we want to subject the enzyme to different temperatures. So for the 0 degrees tube, you would stick this test tube in an ice box for 5 minutes or so. So 5 minutes in an ice chest or ice box. And then after you've basically frozen the conditions or dramatically decreased the temperature, then you'd add your starch. So then 2 mils of starch, just like before. And then, of course, right as you do that, the reaction occurs. You wait the 30 seconds, just like before. Pipette, 30 seconds, pipette, etc. And then you would do this after you'd finish that row. You'd do it for what? Room temperature, which is 20 degrees Celsius, 37 degrees Celsius, 53, and 80. So there's a warm water bath, room temperature, and then there's a hot water bath too. That, again, you would incubate the enzyme for about 5 minutes in the temperature condition so that the enzyme's exposed and becomes that temperature. Then you would add the starch, and then you're testing to see if the enzyme works after being exposed to these different conditions. That's the goal of this lab. And you see that on the well plate. The enzyme works. If it's breaking down starch, you're going to see that color change. Because remember, the Lugol is identifying if starch is present. If it is, it turns blue-black, and the enzyme's not breaking it down, right? If it doesn't change colors, this lab is really backwards and tricky. If it doesn't change colors, then starch did break down and it isn't present. So that's if it doesn't change colors. And so if it doesn't change colors, the enzyme is working. So say your wells don't change colors like at all after doing this at freezing temperatures, then your enzyme still works at those freezing temperatures. It's still breaking down. And you could say, hey, enzyme activity is still great at freezing temperatures. As a hint, that's not really what happens. Enzyme activity will actually slow down, and that's part of why you do this experiment, to discover how these conditions affect enzyme activity and reaction rates. But that's it. That's what you do. Whew. Right on time. All right, so is anyone still there? Does anyone have any questions about this lab? It's certainly a trippy one, but a lot of similarities from last week. So any questions? Do we need the pics of the 96 plate? No, just the only thing you need is what's in the lab packet that I sent you. And the pictures are embedded in the packet, the Google Doc version and the Microsoft Word version. Yeah, you just pick one. Yeah, you can just pick one as far as answering the two. Yeah, because in real life, in person, I would assign a group one condition. Like I'd have you guys do temperature, I'd have you guys do NaCl. So the hypothesis question and the graph, just do that for one factor. But then, of course, like I said, be sure, and it does say everyone must respond to the following. Be sure to answer the question for the results and summarizing the results for all three. And that's after the graph stuff. What do the buffers do? That's a good question. The buffers, well, I think I mentioned this last week and I had to think about it, but the buffer is to resist changes in pH that might affect the enzyme activity. Remember, we're trying to isolate one variable and we don't want pH to influence like when we're testing temperature, right? And the nature of just the tube, the saliva itself might be slightly acidic. The starch maybe. The pH, the buffers by definition resist changes in pH. They absorb hydrogen ions or they release hydrogen ions to neutralize pH changes. They try to keep it the same that the tube is in, kind of thing. So like in our blood, it's like a slightly basic pH above 7. We have bicarbonate buffer. So if any acidic thing enters our blood, the buffer, bicarb, will absorb the hydrogens and prevent it from changing the pH. Does that make sense? Okay. Yeah, you don't have to worry about like why we have the buffer, but it has something to do with maintaining the pH of the, I think saliva is slightly acidic maybe. It's a good question though. I do know that's what buffers do. Okay guys, well, that's it. You have all the lab info. Just let me know if you have any questions. You have the 2 lecture quizzes. This is my synchronous section. You got the 2 lecture quizzes due Friday. That's it. So I'll see you Thursday, okay? Oh, the hypothesis from part 1, I'll answer that. You guys can stick around if you want, but feel free to bail if you'd like as well. So, like I said, I didn't assign you a hypothesis last week, which is totally cool. Just kind of come up with one for how you think the environmental factor will affect the pH, or I mean the enzyme activity. So say you're assigned temperature. A hypothesis could be as temperature increases, enzyme activity decreases. Just come up with one. You could say enzyme activity increases as temperature goes up, as I try increasing the temperature. And the rate of reaction and the starch breakdown will increase or decrease, whatever you come up with. Then you're just gonna look at the data and say, hey, is it true? Does the data support the hypothesis that I just came up with, or reject it? That's all. Remember, a hypothesis is just a guess, an educated guess about your results before finding out what the results are. And so you're seeking out to support it, prove it. We don't say prove, right? Support it or reject it. Other questions? You got it. Okay. Well, again, just email me if anything comes up, and I'll see you Thursday. So have a great rest of your afternoon. Padres, go Padres. All right, guys. You're welcome. Thank you.